Hello students, Jinali Shah welcomes you all on behalf of Saraswat International Academy. Today we will begin with the next part of the chapter Biotechnology and its applications. Students, in the previous video we have studied how biotechnology is applicable in the field of agriculture. In today's lecture, we will see where the biotechnology is applicable in other fields such as medicine, then in biopatenting, etc. So let us begin with our topic, the biotechnological applications in medicine. How is biotechnology applicable in the field of medicine? The recombinant DNA technological processes have made immense impact in the area of healthcare by enabling the mass production of safe and more therapeutic drugs. So basically the recombinant DNA technology, by the use of the recombinant DNA technology, we are able to produce the mass production of safe and in bulk quantity the therapeutics drug. Due to the biotechnology, whatever therapeutic drugs are available, they are produced in large scale as well as they are safe. Further, the recombinant therapeutics do not induce unwanted immunological responses as in common in case of the similar products isolated from non-human sources. ओके क्या बोला गया है कि ये थ्रू द रिकॉम्बिनेंट डीएनए टेक्नोलॉजी जो भी थेराप्यूटिक्स बने हैं, सो दिस थेराप्यूटिक्स डू नॉट इंड्यूस अनवांटेड रेस्पॉन्सेज. सो पहले दो सिमिलर प्रोडक्ट, से फॉर एग्जांपल देयर इज अ प्रोडक्ट, आई से फॉर एग्जांपल इंसुलिन इनिशियली was obtained from the non-human resources, and nowadays through the use of the recombinant DNA technology we are able to produce the human insulin itself. So जो recombinant DNA technology से insulin production हुआ है, that will not create, that won't induce the unwanted immunological responses, and the insulin which is isolated from non-human resources, that insulin will generate some unwanted responses in the human body. So through the use of the recombinant DNA technology, what is happening? Unwanted जो immunological responses generate होते हैं, they won't be generated as they were generated in case of the non-human sources. At present, about 30 recombinant therapeutics have been approved for the human use the world over. Okay, so basically human use the world over. So till now, abhi tak normally 30 recombinant therapeutics have been approved. In India, 12 of these are presently being marketed. So all over the world till now 30 recombinant therapeutics have been approved, and of this 30 approved recombinant therapeutics, 12 are being marketed in our country. So first of all, the genetically engineered insulin. Management of adult onset diabetes is possible by taking insulin at the regular time interval. So इंसुलिन को अगर हमें मैनेज करना है, if we want to control the diabetes, not insulin, sorry students, if we want to control diabetes, then by taking the insulin at regular level, that can be cured. What would a diabetic patient do if human insulin was not available? What would have happened अगर human insulin available नहीं होता तो? So one would have to isolate and use the insulin from the other animals. If human insulin was not available, what was the case? What would have happened? Then one would have to isolate the insulin from the other animals. Would the insulin isolated from other animals be just as effective as that secreted by the human body itself? And would it not elicit an immune response in the body? Okay, अगर human insulin available नहीं होता तो क्या कंडीशन आती कि वी वुड हैव टू आइसोलेट द इंसुलिन फ्रॉम द अदर एनिमल्स. नाउ क्वेश्चन अराइजेस, whether the insulin isolated from the animals would be as effective as the insulin isolated from the human beings. यानी जो एनिमल में से इंसुलिन को आइसोलेट किया गया है, क्या वो इंसुलिन की इफेक्टिविटी ह्यूमन इंसुलिन के जैसी होगी? एंड अदर क्वेश्चन अराइजेस इज, वुड दैट इंसुलिन इंड्यूस सम immune response in the body? Question arises. वो जो इंसुलिन एनिमल में से आइसोलेट करके ले गए, सो वो इंसुलिन कैसा होगा? बेसिकली इट विल बी अ फॉरेन पार्टिकल टू द बॉडी, एंड व्हेन सम फॉरेन पार्टिकल एंटर्स अवर बॉडी, अवर इम्यून सिस्टम इज एक्टिवेटेड एंड इट वुड गिव द इम्यून रिस्पॉन्स टू दैट पर्टिकुलर फॉरेन बॉडी. सो क्या प्रॉब्लम आता? whether there would be whether the effectivity of the insulin isolated from the animal be same as that secreted by the human body itself. And next question is, would it induce some immune response in the human body? So students, of course इसका आंसर yes है. Effectivity would not be the same as the insulin secreted by the human beings. And another thing is, of course it would elicit or it would induce the immune response in the body. वो इंसुलिन easily acceptable नहीं होता क्योंकि our body considers that insulin as a foreign particle. Now suppose, imagine if a bacterium were available that would make the human insulin. Now ऐसा imagine करो कि बैक्टीरिया ही प्रोडक्शन करेगा ह्यूमन इंसुलिन का. कैसे? ह्यूमन इंसुलिन की जो जीन्स है उसको हम आइसोलेट करेंगे, उसका हम रिकॉम्बिनेंट टेक्नोलॉजी से वेक्टर में ऐड करेंगे, एंड देन दैट वेक्टर विल बी अलाउड टू एंटर इनटू द बैक्टीरिया. नाउ जैसे जैसे बैक्टीरिया का मल्टीप्लिकेशन होगा, ह्यूमन इंसुलिन की जीन्स का मल्टीप्लिकेशन होगा, एंड ह्यूमन इंसुलिन की जीन एक्सप्रेस होकर क्या बनाते? ह्यूमन इंसुलिन का प्रोडक्शन करती. Suddenly the process would become so simple. So you can easily grow a large quantity of bacteria and make as much insulin as you need. So another question, whether insulin can be orally administered to the diabetic people or not. क्या इंसुलिन orally ले सकते हैं? Whether the diabetic patients can take insulin orally or not. So the answer to this question is no. इंसुलिन कैसा हार्मोन है? पेप्टाइड हार्मोन है. So if insulin is taken orally, what would have happened? स्टमक में एंड इंटेस्टाइन में जो डाइजेस्टिव जूसेस है वो इंसुलिन को क्या कर देते? ब्रेक कर देते बिफोर दैट इंसुलिन रीचेज द ब्लड स्ट्रीम, एंड हेंस इंसुलिन कैन नॉट बी टेकन ओरली. So no, insulin cannot be administered orally because digestive juices in stomach and intestine will break down the insulin before it reaches the blood stream. So this is the reason why the insulin cannot be taken orally. Now, insulin used for diabetes was earlier extracted from the pancreas of the slaughtered cattle and pigs. So before the recombinant DNA technology, डायबिटीज़ के ट्रीटमेंट के लिए जो इंसुलिन आइसोलेट किया जाता था, इट वॉज आइसोलेटेड फ्रॉम द पेंक्रियाज ऑफ स्लॉटर्ड कैटल्स एंड पिग्स. Now, insulin from animal source though caused some patients to develop allergy or other types of reactions to the foreign protein. अभी ये जो इंसुलिन एनिमल्स का इंसुलिन यूज़ किया जाता था, so that insulin it developed some type of the allergy in the patients and some reactions. Why? Because that insulin was a foreign protein. Now, when we talk about the composition of the insulin consists of two short polypeptide chain. Insulin is made up of two chains, chains A and chain B. Chain A has 21 amino acid and chain B has 30 amino acid. Chain A के पास कितने amino acids है? 21 and B के पास 30. So total 51 amino acids are present in the insulin molecules. Now both the chains are linked by the 2 disulfide bridges. अभी ये दो chains होती है वो linked होती है एक दूसरे से. How? So there are 2 disulfide bonds which join the 2 polypeptide chains of insulin. Now students, insulin जब produce होता है human body में, so normally there are 2 chains, chain A and chain B. But also along with the insulin, chain C is produced and this insulin is called as proinsulin. अभी ये proinsulin इंसुलिन में कन्वर्ट होता है तब द चेन सी विल बी रिमूव्ड. सी स्टूडेंट्स, हियर ब्लू कलर्ड इज चेन ए, ग्रीन कलर इज चेन बी एंड द ग्रे कलर्ड इज द चेन सी. सो जब इंसुलिन इनिशियली बनता है then along with chain A and chain B there is the presence of chain C. And here you can see this is one disulfide bridge and this is the another disulfide bridge. Now ये इंसुलिन को क्या बोलते हैं? प्रोइंसुलिन. Now when the proinsulin is converted into the mature form of insulin, ये प्रोसेस को बोलते हैं maturation. At that time the C polypeptide और C peptide is removed. देखो यहाँ पे ये free C peptide है and the remaining is chain A and chain B which are linked with the disulfide bridges. So this is the maturation of proinsulin into insulin. यानी जब इंसुलिन का synthesis होता है, so along with the chain A and B, chain C is also synthesized and this chain C at the time of the maturation will be removed and only chain A and chain B will be left. So that process is called as the maturation of the proinsulin into the insulin. In mammals, including the humans, insulin is synthesized as a prohormone. Like a proenzyme, the prohormone also needs to be processed before it becomes a fully mature and functional hormone which contains an extra stretch called as the C-peptide. जैसे मैंने बताया कि mammals including the human beings, in them whenever insulin is synthesized, it is synthesized as a prohormone. Prohormone is like a proenzyme. Proenzyme कैसी होती है कि this proenzyme needs to be converted into the active form of the enzyme. Same way, insulin is synthesized as a prohormone and this prohormone also needs to be processed before it is converted into the mature and functional hormone. So ये जो proinsulin होता है which occurs in the form of prohormone, it contains an additional chain which is called as the C-peptide. This C-peptide is not present in the mature insulin and is removed during the maturation into insulin. So जो mature insulin होता है इसमें C-peptide नहीं होता है. It means during the maturation process that C-peptide will be removed. The C-peptide is removed during the conversion of prohormone to mature hormone. The process is called as maturation. That is, the removal of the C-peptide during the conversion of the prohormone to mature hormone is called as the maturation. Now, the main challenge for production of the insulin using a recombinant technique was getting insulin assembled into a mature form. Okay, ये हमारी बॉडी में होता है कि 3 chains are synthesized, यानी पहले prohormone secrete होता है containing peptide A, peptide B and peptide C. The peptide C will be removed during the maturation and its conversion from prohormone to the active form of the hormone. सो रिकॉम्बिनेंट डीएनए टेक्नोलॉजी से जो चैलेंज थी, मेन चैलेंज क्या थी? टू असेंबल द इंसुलिन इन द मैच्योर फॉर्म. So in 1983, Eli Lilly, an American company, they prepared the two DNA sequences corresponding to chains A and B of the human insulin. Eli Lilly company ने क्या किया? So they the genes coded to synthesize the chain A and genes coded to synthesize gene B. So उन्होंने ये दोनों DNA sequences को prepare किया. They prepared the DNA sequences corresponding to chain A and chain B and then introduced them in the plasmids of E. coli to produce the insulin chain. अभी ये दोनों chain को उन्होंने क्या किया? They introduced into the plasmids and then into the E. coli. चेन ए एंड बी वर प्रोड्यूस सेपरेटली. क्यों? क्योंकि हमने दोनों जीन्स को सेपरेट करके प्लाज्मिड में एंटर किए थे एंड देन दिस टू चेन्स वुड प्रोड्यूस सेपरेटली. दिस सेपरेटली प्रोड्यूस्ड चेन वर एक्सट्रैक्टेड एंड देन कंबाइंड बाय क्रिएटेड द डाइसल्फाइड बॉन्ड्स टू फॉर्म द ह्यूमन इंसुलिन. सो ये दो चेन सेपरेटली प्रोड्यूस की गई थी. दिस सेपरेटली प्रोड्यूस्ड चेन्स वर एक्सट्रैक्टेड and then combined by creating the disulfide bond to produce the human insulin. So that is how the therapeutic called as insulin is produced through the recombinant DNA technology. Now how recombinant DNA technology is useful in gene therapy? So gene therapy is a corrective therapy for a person born with a hereditary disease. Suppose if a person is born with the hereditary disease, then one way of curing that particular hereditary disease is gene therapy. What gene therapy indicates? It means to change the damaged or mutated genes and entry of the genes doing the normal functions. What do you mean by gene therapy? सो पर्सन में जो भी डैमेज जीन है या म्यूटेटेड जीन है उसको चेंज करना, उसको रिप्लेस करना. किससे? रिप्लेसिंग इट विद द नॉर्मल जीन्स विच कैरी आउट द नॉर्मल फंक्शंस. जीन थेरापी इज अ कलेक्शन ऑफ मेथड्स दैट अलाउज the collection of a gene defect that has been diagnosed in a child or a embryo. So if a child or the embryo of the child is detected with some defective genes, then gene therapy is a method which allows the correction of that defective genes. Here genes are inserted into the person's cells and tissues to treat the disease. So genes are inserted into the person's cells or tissues to treat the disease. So जो भी normal genes है that will be inserted into the cells of the person. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. So how is it done? So here there will be the delivery or entry of a normal gene into the cell or embryo, and that normal gene, it takes over the function of the non-functional gene. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. So the first gene therapy was done in 1994 and it was given to a 4-year girl who was suffering from ADA, that is adenosine deaminase deficiency. Now ये जो जीन है बेसिकली ये एंजाइम है. ये जो एंजाइम है इट इज़ वेरी वेरी इम्पॉर्टेंट फॉर द इम्यून सिस्टम टू फंक्शन. अगर ये जीन्स फंक्शनल नहीं है तो लिम्फोसाइट्स का जनरेशन प्रॉपर होगा नहीं, एंड लिम्फोसाइट्स प्रॉपर नहीं यानी इम्यून सिस्टम डिफेक्टिव रहेगी. So a person with ADA deficiency will have the immunological problems. This enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of the gene for the adenosine deaminase. Okay, ये जो deficiency है इसमें क्या होता है? So basically the gene which is responsible to secrete ADA, that is adenosine deaminase, it is deleted from the person. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy, in which the functional ADA is given to the patient by injection. So initially क्या किया जाता था कि suppose bone marrow transplantation करते हैं तो that deficiency can be removed. Another way is to give the person the functional ADA injections. But problem with both this technique was that they are not completely curative. ये दोनों approach, that is bone marrow transplant and to give the functional ADA injections, ये दोनों technique completely curative नहीं है. So as a first step towards gene therapy, lymphocytes from the blood of the patient are grown in a culture outside the body. So what is done in this technique? The lymphocytes from the patient bloods, they are grown in the culture outside the body. A functional ADAC DNA, that is complementary DNA using a retroviral vector. Animals के लिए mainly vectors कौन होते हैं? Retroviruses, disarmed retroviruses. It is then introduced into these lymphocytes which are subsequently returned to the patient. Complementary DNA is a DNA synthesized from the single-stranded RNA template in a reaction catalyzed by the enzyme reverse transcriptase. It is often used to clone the eukaryotic genes in the prokaryotes. However, as these cells are not immortal, अभी lymphocytes जो culture की है वो immortal तो होने वाले नहीं है. The patients require periodic infusion of genetically engineered lymphocytes. So ये जो culture किए गए cells immortal तो होने वाली नहीं है. So periodically this genetically engineered lymphocytes need to be given to the patient. But if the gene isolated from the marrow cells producing ADA is introduced into the cells at early embryonic cells, it would be a permanent cure. So जो gene ADA प्रोड्यूस करते हैं वो हम मैरो में से आइसोलेट करके एम्ब्रियोनिक स्टेजेस में ही वो अगर सेल्स इंट्रोड्यूस कर दे then it would be a permanent cure to the disease. See how it is done. The SCID, that is severe combined immunodeficiency patient, has a defective gene for enzyme adenosine deaminase. He or she lacks the functional T lymphocytes and fails to fight the infective pathogens. Lymphocytes are extracted from the patient's bone marrow and a normal functional copy of a human gene coding for ADA is introduced into these lymphocytes with the help of retrovirus. क्या करते हैं? बोन मैरो में से lymphocytes को extract करते हैं and then the functional gene which codes for the enzyme ADA is introduced in these lymphocytes using the retrovirus. The cells so treated are reintroduced into the patient's bone marrow. Okay, फिर से ये cells कहाँ पे डाल देते हैं? Bone marrow में. Now what will happen? The lymphocytes produced by this cell contains functional ADA genes and reactivates the victim's immune system for life. See students, यहाँ पे ये complete technology दी गई है. The bacterium carrying a plasmid with a cloned normal ADA gene. This is a plasmid which contains the ADA gene and this is a genetically disabled retrovirus के जिसमें से pathogenicity remove की गई है. Now ये process कैसे करते हैं? The cloned ADA gene is incorporated into virus. बैक्टीरिया जिसमें प्लाज्मिड होता है, ये प्लाज्मिड के पास कौन सी जीन होती है? ADA gene. This will be introduced into a virus. Now, the T cells with the disabled ADA gene. Now, जो पर्सन को स्किड पेशेंट है जिसमें ADA डेफिशिएंसी है उसके लिम्फोसाइट्स को आइसोलेट करते हैं. ओके, नाउ एंड दिस इज व्हाट कि ये जो क्लोन्ड ADA जीन वाला जो प्लाज्मिड है उसको हम कहाँ पे इंट्रोड्यूस करते हैं, रेट्रोवायरस में. नाउ द रेट्रोवायरस विल इन्फेक्ट द T cells and will transfer the ADA genes into the cell. So आप देख सकते हो कि ये lymphocytes है जिसमें ये genes क्या हो गई है, एंटर हो चुकी है. Now the cells are grown in the culture to ensure that the ADA gene is active, and then these genetically altered cells are replanted to produce the ADA. So this is how this technique is carried out. Now the use of the gene therapy in molecular diagnosis. We know that effective treatment of a disease, for an effective treatment of disease, early diagnosis and understanding its pathophysiology is important. कोई भी डिजीज़ की इफ़ेक्टिव ट्रीटमेंट के लिए क्या ज़रूरी है, फर्स्ट थिंग रिक्वायर्ड इज़ अर्ली डायग्नोसिस ऑफ द डिजीज़ सो दैट disease does not spread much more in the body, and second one is to understand the pathophysiology of that disease. ये disease कैसे spread करता है, वो क्या डिफेक्ट करता है, उसका स्टडी. Using conventional methods of diagnosis, that is serum and urine analysis, early detection is not possible. Okay, अभी जो conventional normal techniques है, तो normally हम अभी जो techniques यूज़ होती है उसमें क्या करते हैं, we basically carry out blood and urine analysis and through that the disease diagnosis is done. But इससे early detection नहीं होगा क्योंकि already ये अपनी body में disease spread हो चुका होगा. Recombinant DNA technology, polymerase chain reaction and enzyme linked immunosorbent assay, that is ELISA, are some of the techniques that serve the purpose of early diagnosis. Presence of pathogen, that is bacteria, viruses, etc., is normally suspected only when pathogen have developed a disease symptom. Okay, conventional method से जो हम diagnosis करते हैं ये कब possible होगा, जब pathogen ने already कोई symptoms produce किए होंगे. By this time the concentration of pathogen is already very high in the body. Symptoms produce किए हैं, इसका मतलब क्या हुआ कि pathogen का concentration it is very high in the patient. However, very low concentration of bacteria or virus at the time when symptoms of the disease are not yet visible can be detected by amplification of their nucleic acid by PCR. सो अगर लो कॉन्सेंट्रेशन बैक्टीरिया का या वायरस का पैथोजन का बॉडी में कम होता है, कब कम होगा कि जब तक सिम्टम्स आए भी नहीं है. ये टाइम पे अगर हम न्यूक्लिक एसिड्स को PCR की टेक्निक से एम्पलीफाई करेंगे देन अर्ली डायग्नोसिस कुड बी पॉसिबल. PCR can detect very low amounts of DNA by amplifying that piece of DNA to form many copies. PCR is now routinely used to detect the HIV in suspected AIDS patients. It is being used to detect the mutations in genes in suspected cancer patients too. So these are the applications of PCR in various fields. It is used in the detection of HIV. It is also used in the detection of the cancer. It is a powerful technique to identify many other genetic disorders. A single-stranded DNA or RNA tagged with a radioactive molecule is allowed to hybridize to its complementary DNA in a clone of cells followed by the detection using autoradiography. So कैसे करना है ये हमने already study किया है. A single-stranded DNA or RNA which will be tagged with the radioactive molecule, it is allowed to hybridize with the complementary DNA and the clone of the cells will be then detectable through the autoradiography. The clone having the mutated gene will hence not appear on the photographic film. अभी जो भी म्यूटेटेड जीन होगी वो फोटोग्राफिक फिल्म में दिखेगी नहीं, बिकॉज वो जो प्रोब है कॉम्प्लीमेंट्री डीएनए का उसने वो पर्टिकुलर पार्ट से अटैच नहीं किया होगा, बिकॉज द प्रोब विल not have complementarity with the mutated gene. म्यूटेटेड जीन होगा तो वो प्रोब और म्यूटेटेड जीन के बीच में कॉम्प्लीमेंटरिटी नहीं होगी एंड हेंस it won't be visible on the photographic film. ELISA is based on the principle of antigen-antibody interaction. Infection by pathogens can be detected by the presence of the antigens, which are proteins or glycoproteins, or by detecting the antibodies synthesized against the pathogen. So ELISA is an antigen-antibody reaction कि जहाँ पे we can detect the presence of the antigens or also the presence of the antibodies which are synthesized against that pathogens. Now, transgenic animals. Animals that have had their DNA manipulated to possess and express an extra foreign gene are known as transgenic animals. So, what are transgenic animals? Transgenic animals are those animals के जिसका DNA manipulate किया गया है. कैसे? By the entry of the extra gene and expression of that gene. से फॉर एग्ज़ाम्पल कोई एनिमल पर्टिकुलर प्रोडक्ट बना नहीं रहा है. से फॉर एग्ज़ाम्पल काउज़ है, काउज़ तो ह्यूमन मिल्क का प्रोडक्शन नहीं करेगी. ओके, नाउ अगर काउ के अंदर हम ह्यूमन मिल्क जो जीन बना रही है वो जीन काउ के अंदर डाल दे तो हमने काउ को क्या किया है, काउ के जेनेटिक कंस्टिट्यूशन को मैनिपुलेट किया है. So that cow is now called as what? Transgenic cow. So, transgenic rabbits, pigs, sheep, cows and fish have been produced, although over 95% of all existing transgenic animals are mice. Now we have transgenic rats, rabbits, pigs, sheep, cows and fishes. But maximum amount में जो transgenic animals है 95% of the transgenic animals are mice. Now question arises, why are these animals being produced? How can man benefit from such modifications? So question arises कि पहले तो क्यों transgenic animals बनाते हैं and are there some or is there some benefits by producing such modification? So let us try and explore some of the reasons. So normal physiology and development. First reason is normal physiology and development. Transgenic animals can be specifically designed to allow the study of how genes are regulated and how they affect the normal functions of the body and its development. Okay, transgenic animal का क्या use हो सकता है? We can study how the genes are regulated, how the genes can be regulated, and second, how that genes affect the normal functioning of the body and its development. Study of complex factors involved in growth such as insulin-like growth factors. So how insulin growth factor is involved in the process of growth. ऐसा कुछ हमें study करना है कि how certain factors are responsible for growth. So उसको study करने के लिए transgenic animals are used. By introducing the genes from the other species that alter the formation of this factor and studying the biological effects that results, information is obtained about the biological role of the factor in the body. So अगर say for example insulin like growth factor की genes हमने कोई दूसरे species में enter की, so now that species will form that factor and we can study the effects which results from the addition of that factors. The next one is the study of the disease. Many transgenic animals are designed to increase our understanding of how genes contribute to the development of disease. So transgenic animals are also designed to understand how genes are responsible for the development of certain diseases. These are specially made to serve as models for human diseases so that investigations of new treatment for the disease is made possible. क्यों ये transgenic animals बनाते हैं? To study the disease. The reason is these animals they serve as the model for human disease. So क्या कर सकते हैं हम? Investigations कर सकते हैं for the treatment of the diseases. डिजीज़ कैसे स्प्रेड हो रहा है, क्या डिफेक्ट कर रहा है, वो हम स्टडी करके वो डिजीज़ को कैसे ट्रीट किया जाता है, दैट कैन बी मेड पॉसिबल. Today, transgenic models exist for many human diseases such as cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's. ये सब disease के लिए transgenic models are available. The next one is the biological products. Medicines required to treat certain human diseases can contain biological products, but such products are often expensive to make. Okay, जो medicines हम लोग certain disease के लिए use करते हैं, that medicines contain biological products, but ये जो products होते हैं वो medicine को बहुत expensive बना देते हैं. Transgenic animals that produce the useful biological products can be created by introduction of the portion of the DNA or genes which codes for a particular product such as human protein alpha-1 antitrypsin which is used to treat emphysema. अभी क्या कर सकते हैं? मेडिसिन्स में जो बायोलॉजिकल प्रोडक्ट्स होते हैं वो मेडिसिन को एक्सपेंसिव बना देती है, तो उसके जगह पर हम क्या कर सकते हैं कि ये बायोलॉजिकल प्रोडक्ट जो जीन बनाते हैं, दैट जीन कैन बी इंट्रोड्यूस्ड इनटू द एनिमल्स एंड नाउ animals will have the genes which will code for that biological product. That biological product could then be isolated and can be used in the treatment of the disease. एक एग्जाम्पल दी गई है कि अल्फा-1 एंटीट्रिप्सिन इट इज़ अ ह्यूमन प्रोटीन. ये प्रोटीन सिर्फ ह्यूमन बींग्स बनाते हैं. सो ये प्रोटीन जो जीन बनाती है वो जीन को आइसोलेट करके ट्रांसजेनिक एनिमल्स में हम डाल देंगे. So now that animal will also start the production of antitrypsin. And what is the usefulness of antitrypsin? It is used to treat the diseases like emphysema. Similar attempts are being made for the treatment of phenylketonuria and cystic fibrosis. In 1997, the first transgenic cow, Rosie, produced human protein enriched milk. Okay, 1997 में रोजी काउ, it is a first transgenic cow, and that cow it produced the milk which was having the human protein, and the quantity of that was 2.4 gram per liter. The milk contained the human alpha-lactalbumin and was nutritionally a more balanced product for human babies than the natural cow milk. ओके, वो जीन इंट्रोड्यूस की गई. सो काउ ने ही किसका प्रोडक्शन किया? ह्यूमन अल्फा लैक्टाल्ब्यूमिन, एंड दिस मिल्क वाज मोर बैलेंस्ड कंपेयर्ड टू द नेचुरल काउ मिल्क. वैक्सीन सेफ्टी. Transgenic mice are being developed for use in testing the safety of the vaccine before they are used on humans. कोई भी वैक्सीन्स नॉर्मली डायरेक्टली ह्यूमन पे ट्राई नहीं करते हैं, उनको एनिमल्स पे ट्राई करते हैं. सो नॉर्मली देयर आर ट्रांसजेनिक माइस के जिसके ऊपर वैक्सीन की सेफ्टी चेक करते हैं. ट्रांसजेनिक माइस आर बीइंग यूज्ड टू टेस्ट द सेफ्टी ऑफ द पोलियो वैक्सीन. इट सक्सेसफुल एंड फाउंड टू बी रिलायबल. They would replace the use of the monkeys to test the safety of the batches of the vaccine. Okay, अगर ये mice reliable है, if these mice are reliable and ये अगर successful हुआ तो we can replace mice with the monkeys. Initially ये testing monkeys पे किया जाता था, तो वो हम avoid कर सकते हैं. Chemical safety testing. This is known as toxicity or safety testing. The procedure is same as that used for testing the toxicity of the drugs. Transgenic animals are made that carry the genes which make them more sensitive to toxic substance than non-transgenic animals. So animals are designed in such a way that the animals carry the genes which make them more sensitive to toxic substance than the non-transgenic animals. Later they are exposed to the toxic substances and effects are studied. Okay, बाद में वो toxic substance से वो animal को expose किया जाएगा and what are the effects that will be studied. Toxicity testing in such animals will allow us to obtain the results in the less time. Now there are certain ethical issues created due to the transgenic models of the animals. क्या ethical issues हो सकता है? So here we will study GEAC and its function. Now what is meaning of ethical? Meaning of ethical is doing right things for the society and unethical means doing harm to the society. Ethical यानी society के लिए कुछ अच्छा करना and unethical it means doing harm to the society. Now through transgenic animals हम क्या कर रहे हैं? Animals के genetic normal composition को change करके उनको हम दूसरी जीन्स ऐड करके उनको मैनिपुलेट कर रहे हैं. सो क्या ये चीज़ प्रॉपर है? एनिमल्स अपने फ़ायदे के लिए एनिमल्स को यूज़ करना, इज़ दैट एथिकल और नॉट? द मैनिपुलेशन ऑफ द लिविंग ऑर्गेनिजम्स बाय ह्यूमन रेस कैन नॉट गो एनी फर्दर विदाउट रेगुलेशन. अगर कोई रूल्स एंड रेगुलेशन नहीं है तो ये प्रॉपर चीज़ नहीं है. Some ethical standards are required to evaluate the morality of all human activities that might help or harm the living organisms. Going beyond the morality of such issues, the biological significance of such things is also important. Genetic modification of organisms can have unpredictable results when such organisms are introduced into the ecosystem. कोई भी ऑर्गेनिजम्स के जेनेटिक मॉडिफिकेशन किया गया है, देन दे विल हैव सम अनप्रिडिक्टेबल रिजल्ट्स व्हेन दिस ऑर्गेनिजम्स आर अलाउड टू एंटर द इकोसिस्टम. Some agricultural companies have started making genetically modified crops that did not produce viable seeds. This forced the farmers to produce new seeds for each season. The only solution was to put some regulatory measures on this activity, taking the care of mortality of such issues on one hand and the biological importance of these studies on other. अभी क्या प्रॉब्लम आया कि सर्टन कंपनीज ने जेनेटिकली मॉडिफाइड क्रॉप्स को मार्केट कर दिया था एंड ये क्रॉप्स ने सीड्स का प्रोडक्शन नहीं किया. इसका मतलब ये हुआ कि फार्मर्स को एवरी सीजन न्यू सीड्स को परचेज करना पड़ा. सो ये सब चीज़ को स्टॉप करने के लिए द सोल्यूशन गिवन वॉज टू हैव सम रेगुलेटरी मेजर्स एंड ऑल्सो टू स्टडी द बायोलॉजिकल इम्पोर्टेंस इन दिस स्टडीज. Therefore, the Indian government has set up the organization such as GEAC, that is Genetic Engineering Approval Committee. यानी जो भी मैनिपुलेशन करते हैं एंड उसका स्टडीज़ करके कोई भी प्रोडक्ट को कमर्शियलाइज करना है तो GEAC का अप्रूवल इम्पॉर्टेंट है. दे विल मेक द डिसीजंस रिगार्डिंग द वैलिडिटी ऑफ द GM रिसर्च. कोई रिसर्च करना है तो भी उनकी परमिशन लेनी है and also the safety of introducing the genetically modified organisms for public services. Now, let us study biopatenting. Biopatent is a government protection to an inventor of the biological material, securing to him for a specific time the exclusive right of the manufacturing, exploiting, using and selling of an invention. So biopatent, patent क्या होता है? The patent is given by government to the inventor of particular product कि उसकी वो प्रोडक्ट की जो भी कोई राइट्स है वो इन्वेंटर को स्पेसिफिक टाइम के लिए मिलेगी. So when patent is given to biological products, it is called as the biopatenting. So what is biopatent? It is a government protection given to an inventor of the biological material, securing him for the specific time the exclusive right of the manufacturing, exploiting, using and selling an invention. The modification or usage of living organisms for public services as food and medicine resources, for example, has also created problems with patents granted for the same. Okay, living organisms का हम use करते हैं to produce, say for example, living organisms are used as a sources of food and medicine etc. और उसका अगर patenting करेंगे so that have created problem. कैसे? There is a growing public anger that certain companies are being granted patents for the products and technologies that make the use of the genetic materials, plant and other biological resources that have been long identified, developed and used by the farmers and indigenous peoples of the specific region or country. Okay, क्या बोल रहे हैं कि देर आर सर्टन प्लांट्स, एनिमल्स या कोई भी बायोलॉजिकल रिसोर्सेज है जो पहले से ही उसका स्टडी की गई है, उसकी बेनिफिट्स क्या है, उसका यूज़ कैसे किया गया है, ये नॉलेज किसके पास है? फार्मर्स या पीपल विच आर इंडिजीनियस और नेटिव टू दैट कंट्री. से फॉर एग्जाम्पल मैं नीम की बात करती हूँ. सो नीम एज अ मेडिसिन, नीम कैन बी यूज्ड एज अ मेडिसिन, इट हैव द एंटीसेप्टिक इफेक्ट्स. ये जो ट्रेडिशनल नॉलेज है वो अपने पास है. नाउ व्हाट द बिगर कंपनीज डू, द मल्टीनेशनल कंपनीज अपने नॉलेज को क्या करते हैं? एक्सप्लॉइट करते हैं एंड उसका पेटेंटिंग करवा देते हैं. सो क्या होता है? नाउ नीम का एज अ मेडिसिनल यूज़, उसका जो पेटेंटिंग है, उसका जो बेनिफिट्स मिलने वाले हैं वो किसको मिलेगा? जिसको ये पेटेंट दिया गया है. सो व्हाट बिग कंट्रीज डू, डेवलप्ड कंट्री के पास मनी है लेकिन उनके पास ट्रेडिशनल नॉलेज मेनली नहीं होता है. एंड जो डेवलपिंग कंट्रीज होते हैं उनके पास ट्रेडिशनल नॉलेज बहुत ज़्यादा है, बट ड्यू टू द लैक ऑफ रिसोर्सेज, फाइनेंशियल रिसोर्सेज, वो उसको पर्टिकुलरली पब्लिश नहीं कर सकते. सो व्हाट द डेवलप्ड कंट्री डूज, दे टेक द पेटेंटिंग ऑफ दिस पर्टिकुलर प्रोडक्ट्स एंड देन अपने प्रोडक्ट के नाम से दे विल बी मार्केटिंग दैट पर्टिकुलर प्रोडक्ट. एग्जाम्पल दूसरी, rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. There are an estimated 2 lakh varieties of rice in India alone. The diversity of rice in India is one of the richest in the world. Basmati rice is distinct in its unique aroma and flavor, and 27 documented varieties of basmati are grown in India. There is a reference to basmati in ancient texts, folklore and poetry as it has been grown for centuries. Okay, basmati जो राइस है, there are lakhs of varieties of rice. उसमें जो बासमती है इट इज़ यूनिक फॉर इट्स एरोमा एंड फ्लेवर एंड अपनी कंट्री में 27 वराइटीज़ ऑफ बासमतीज आर ग्रोन. नॉट ओनली दैट, बासमती का रेफरेंस एंशियंट टेक्स्ट में, पोएट्रीज में भी किया गया है बिकॉज इट हैज़ बीन ग्रोन फ्रॉम सेंचुरीज. In 97, an American company got the patent right on basmati rice through the US Patent and Trademark Office. अभी 1997 में क्या किया, एक अमेरिकन कंपनी ने बासमती राइस पे पेटेंट खरीद लिए. इसका मीनिंग क्या हुआ कि बासमती के प्रोडक्शन से लेकर जो भी इनकम से लेकर कुछ भी बेनिफिट्स मिलेगी ये कौन सी कंपनी को मिलेगी? ये अमेरिकन कंपनी को मिलेगी. So this allowed the company to sell a new variety of basmati in US and abroad. This new variety of basmati has been actually derived from the Indian farmer varieties. US ने एक पेटेंटिंग किया एंड देन एक न्यू वैरायटी ऑफ बासमती जनरेट करके उसको सेल किया US में एंड एब्रोड में. लेकिन ये जो न्यू वैरायटी है एक्चुअली वो किस में से प्रोड्यूस की गई है? इट हैव बीन डेवलप्ड फ्रॉम द इंडियन फार्मर्स वैरायटीज ओनली. इंडियन बासमती वॉज क्रॉस्ड विद द सेमी डार्फ एंड क्लेम्ड एज एन इन्वेंशन और नोवेल्टी. क्या किया? इंडियन जो बासमती राइस है उसको उन्होंने किसके साथ क्रॉस किया? सेमी ड्वार्फ के, जिसके ग्रेन साइज कम है, उसके साथ क्लोनिंग करके उन्होंने क्या किया कि ये नई वैरायटी है ऐसा करके उन्होंने बोला. द पेटेंट एक्सटेंड्स टू फंक्शनल इक्विवेलेंस इम्प्लाइंग दैट अदर पीपल सेलिंग बासमती राइस कुड बी रिस्ट्रिक्टेड बाय द पेटेंट. अभी ये पेटेंट में ऐसा था कि कोई दूसरा पर्सन भी बासमती को सेल करेगा तो उसकी पूरी राइट्स वो पर्टिकुलर कंपनी के पास थी. Several attempts have also been made to patent uses, products and processes based on the Indian traditional herbal medicines like turmeric and neem. Now patenting सिर्फ products का नहीं, patenting वो particular products के use कैसे कर सकते हैं, वो processes का भी patenting हो सकता है. So Indian traditional herbal medicine का भी उन्होंने patenting करवा लिया. If we are not vigilant, or you can say careful, and we do not immediately counter these patent applications, other countries or individuals may encash on our rich legacy and we not be able to do anything. अगर हम केयरफुल नहीं रहे और ये सब चीज़ में ध्यान नहीं लिया तो क्या करेंगे, दूसरे कंट्रीज़ या इंडिविजुअल्स अपने रीच नॉलेज को दे विल टेक इट and we won't be able to do anything. Last topic, biopiracy. Biopiracy is a theft or robbery of the biological and genetic resources indigenous to the country. So basically robbery or theft of the biological or genetic resources which are native to the country. ऐसे products है जो वो particular country के पास ही है, कहीं और अवेलेबल नहीं है, एंड इसकी रॉबरी को क्या बोलते हैं, बायोपायरेसी. Biopiracy is a term used to refer to the use of bioresources by multinational companies and other organizations without proper authorizations from the country and people concerned without the compensatory payment. So what is biopiracy? The use of the bioresources by multinational companies and other organizations without the authority from the कंट्रीज एंड पीपल कंसर्न. ओके, कोई बायो रिसोर्स है वो पर्टिकुलर कंट्री से ही अवेलेबल है. सो क्या ये व्हाट इज़ इट, ये रिसोर्सेज को मल्टीनेशनल कंपनी या दूसरी कंपनी क्या कर देते हैं, वो जहाँ पे वो पर्टिकुलर प्रोडक्ट बना है उसके ऑथोरिटी बिना उसका यूज़ कर देते हैं विदाउट गिविंग द पेमेंट. दैट इज़ कॉल्ड एज द बायोपायरेसी. Most of the industrialized nations are rich financially but poor in biodiversity and traditional knowledge. As I told you कि जो developed country है, industrialized nations है, they are rich financially but उनके पास जो traditional knowledge है that is very poor. Vice versa to that, that is in contrast, the developing and the underdeveloped world is rich in biodiversity and traditional knowledge related to bioresources. So जो developing countries है या underdeveloped countries है, though financially they are not rich but they are very rich in their biodiversity as well as the traditional knowledge related to their resources. Traditional knowledge related to bioresources can be exploited to develop the modern applications and can also be used to save time, effort and expenditure during their commercialization. Okay, जो भी ट्रेडिशनल नॉलेज है पर्टिकुलर रिसोर्सेज का, वो क्या करते हैं, वो ट्रेडिशनल नॉलेज को एक्सप्लॉइट करके मॉडर्न एप्लीकेशंस में यूज़ कर देते हैं. से फॉर एग्जांपल द यूज़ ऑफ द नीम एंड टर्मरिक इन द फील्ड ऑफ मेडिसिन. ऑलरेडी हमारे जो एंशियंट पीपल्स थे, हमारे जो एंसेस्टर्स थे, दे हैव ऑलरेडी यूज द मेडिसिन. नीम एंड टर्मरिक का उनको मालूम था कि कैसे उसका मेडिसिनल यूज़ हो सकता है. सो क्या करते हैं, ऑलरेडी इसका प्रोसीजर मालूम है, सो इट वुड हैव सेव टाइम एंड एक्सपेंडिचर ड्यूरिंग द कमर्शियलाइजेशन. देयर हैज़ बीन ग्रोइंग रियलाइजेशन ऑफ इनजस्टिस, inadequate compensation and benefit sharing between the developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. So, क्या कर सकते हैं, डेवलप कंट्री के पास पैसे तो है, तो ये अपने ट्रेडिशनल नॉलेज को खरीद कर क्या कर सकते हैं, यूज़ करके उसको कमर्शलाइज करते हैं, एंड दे डू नॉट टेक द परमिशंस ऑफ द डेवलपिंग कंट्रीज. सो व्हाट टू अवॉइड दिस, the Indian Parliament has recently cleared the second amendment, that is change, of the Indian patent bills that takes such issues into consideration, including patent terms, emergency provisions, research and development initiative. So that is regarding the biopiracy. So we come to the end of this chapter. Thank you students.